Okay, so like I said, having a single base added or lost throws off the entire reading frame. So the parts that the RNA knows that has to be read, and that's called a frame shift mutation. Okay, so to kind of look at an example. So let's say this entire, sequence here was added. Okay. So this is our DNA sequence, right? So we have, we're looking specifically at these two G's. Okay. So if we're splitting it off because our DNA sequences and RNA sequences, when read, are read in sets of 3, so like groups of 3. So 3 amino acids, or an amino acid is coded for by 3 bases. So these are the 3 bases that are together, and then these are the amino acids that are coded for by those bases grouping together. Okay, so let's say that we added a G. Okay, so we added a guanine base in here. Okay, what that means is going to happen is our pairings are going to be messed up because look, this U is now by itself. So now that's going to change our amino acid sequence because there are specific codons. So codons are the word used for those groups of 3 bases. There are specific codons that say stop. Okay, so that way it knows, okay, we cannot continue to have any more amino acids. This is the end of the protein. So when that happens, that's it. The protein gets cut short. Now, if that's the case, then there's a mutation and something's wrong. Okay, so we can have these things called snRNPs. Those are small nuclear RNA. Okay, obviously they're proteins. Okay, then we can have a spliceosome, which is several of those small nuclear RNA. So they recognize the splice site sequence. So they're the cut and paste gene. So they kind of come in and cut and paste and fix that. Alternative mRNAs can be produced from the same gene. That happens when an intron is not actually an intron. So there are different segments that are treated for as exons. So this is more post-transcriptional processing. We need to protect the mRNA on its trip from the nucleus to the cytoplasm in finding that ribosome to become translated into into a protein. So the enzymes in the cytoplasm attack the mRNA. So they have to protect the end of the molecule. So you add a 5' GTP cap. When we say 5' GTP cap, that's the 5' end, guanine and triphosphate, okay? And then you add a poly A tail. So that's just at the 3' end. It's a longer tail. That means the mRNA is going to last longer and it produces more protein. So when we say A, it's adenine. So let's go through this process. How does mRNA code for protein? So we know for DNA to mRNA, all we're doing is translating. So we're just putting it in RNA's language. Okay, so T to A to U, C to G, G to C, etc. Now we have to figure out how are we going to get this into the actual protein. Okay. So how can you go code for 20 different amino acids with only 4 nucleotide bases? Because in total, there are 20 amino acids that make up our proteins. So mRNA codes for proteins in triplets. So this is what I was saying about the specific codons. Okay, so the codons are those groups of 3 different bases. Okay, so since we have only 4 bases, but we can have so many different combinations of them that is how we get the different 20 amino acids. So cracking the code. Okay, so crick this dude, determined a 3-letter triplet or codon system. Okay, so why did the red bat eat the fat rat? Okay, so this is kind of like a way for you to read it. So you group it in the 3s for the codon system. So then you had Nuremberg and Corona, that determined the mRNA amino acid matches. Okay, so they recognize that a codon matches or codes for a specific amino acid. So they tested for that, and they found that the triple U coded for phenylalanine, which is an amino acid. Okay, so this is the code. So this is our codon chart to see whenever you get your DNA sequence and then your mRNA sequence, you can match up your codons to the specific amino acid. Okay, so it codes for all of life. This chart right here codes for all of life. Okay, it's redundant because several codons for each amino acid. So, I mean, you can see all the different codons for the different amino acids. The 3 letters to the side are just the abbreviations for those amino acids. So we can have our start codon. So obviously you can see this is start. We only have 1. So if you see AUG, you know that that is going to be methionine as an amino acid, and that is your start codon. Now you can have 3 different stop codons. You can have UAA, UAG, UGA. Okay, those are your stop codons notifying you, stop, this is the end of the protein. So how are these codons matched to the amino acids? So DNA is 3' to 5'. The mRNA is going to be 5' to 3'. We're going to get our codons. Okay, so our codons here, we're just grouping them into 3s. Okay, now once we group them into 3s, we're going to have an anticodon. The anticodon is basically the opposite of the codon, so the matching of the codon. Because in mRNA, we have it from a 5' to 3', so the anticodon for transfer RNA is going to be the 3' to 5'. Okay, hold on, let me go back. So the transfer RNA is what's going to take that anticodon and attach the amino acid to it. So the transfer RNA structure, okay, and they call it a clover leaf structure. I think it looks more so kind of like a bobby pin type of thing. So anticodons on the clover leaf end, and the amino acid is attached to the 3' end. Okay, so here you can kind of see the anticodon is like that teal part, okay? So at the kind of bubble is the anticodon, and then at the other end, or opening almost, is the amino acid being attached to it. So to actually load the tRNA, you have aminoacyl tRNA synthetase. It's ending in ACE, so you know that it's an enzyme. So it bonds the amino acid to the tRNA. So obviously to bond things, you're going to need some energy, right? So we have ATP converting into AMP. So when we release those 2 phosphates to get to just the monophosphate, energy is being released. So that means the bond is very unstable. So it can release the amino acid at the ribosome very easily, and that's what you want. So the ribosome itself facilitates the coupling of your tRNA anticodon to your mRNA codon. So the structure is going to be looking like this. So you have 2 subunits, okay? So you have a large and a small. Basically what happens is that the mRNA travels through, and the ribosome kind of like almost like a business supply chain type of thing. Like, it's really hard. I'm doing a lot of movements with my hands trying to show you guys. But it's like, it's almost like pushing, the parts together. But, what happens is the mRNA runs through the center of it, and within, the ribosome itself, that's where we're going to be like attaching all of the different parts. Okay, so you have the A site, and the A site is holding the tRNA that's carrying the next amino acid to be added to the chain. Okay, then you have the P site, which is where it's holding the tRNA carrying the growing polypeptide chain. Polypeptide is protein. Okay, and then the E site, which is the exit site. Okay, so the A site kind of is just like where, all right, this is the next amino acid that's going to be added. This is the next one. This is the next one. The P site is the part that's holding on to the protein that's already being built. And the E site is where the tRNA is going to leave the ribosome from the exit site. So it's going to leave it from there. Okay, so this is kind of animations for you to watch if you want to kind of explain this process. So to actually build a polypeptide, so we have initiation. We're going to bring together the mRNA. Now remember, this process is called translation, okay? We are actually translating the RNA sequence into protein language to make a protein. So initiation brings together mRNA, the ribosome subunits, and the initiator tRNA. Initiator tRNA is obviously the tRNA that's going to start the whole thing off. Then we have elongation. Elongation is the process where you're adding the amino acid sequence base, or the amino acids based on the codon sequence. Okay, and then we have termination, where we reach that stop codon and we're ending. Okay, so here's the full story of a gene. There's a YouTube video for you to watch to help kind of break down the process again. Okay, so we have DNA, RNA polymerase. Remember, DNA is replicated in the nucleus. Then DNA cannot leave the nucleus, so RNA has to come in and copy it. Okay, so transcribe it into RNA format. So you have RNA polymerase that comes in to get mRNA. Okay, we have our exons and our introns. We gotta make sure we keep the gene and get rid of the other junk. Okay, then we gotta add those caps on. So the 5' GTP cap and then the poly A tail for the 3' end. So that way we're nice and protected. Then it's actually considered mature mRNA. It travels through the cytoplasm and finds the ribosome. Okay. And then it kind of gets sandwiched in between the two. And that's when we have our A site of our ribosome that comes in and actually starts it all and it brings on the next amino acid. Then you have your P site that's just holding on to the growing polypeptide chain. Okay, and then the E site is where like, okay, we've reached that stop codon, we're Gucci, this is the end of our protein. Okay? There we go. Okay, so these are the transcriptional units. This kind of looks really chaotic, but I mean, if you go through each one, it just kind of like makes a sense for you. Okay, hopefully it does. There's a transcriptional unit, so that's actually the part of the DNA that's transcribed. Okay, then once that is transcribed, we have RNA that comes through and actually RNA polymerase that comes through and actually, you know, makes that mRNA unit. Okay. Now, that is protein synthesis in our eukaryotic cells. Okay. So in the next part, I'm going to do protein synthesis in prokaryotic cells.